Today we're going to get into the meat and potatoes of metabolism. Fast glycolysis, also known as anaerobic glycolysis, is a metabolic pathway that plays a crucial role in providing energy to cells, especially in situations where oxygen is limited or not readily available. This process involves a series of chemical reactions that convert glucose into pyruvate, ultimately generating adenosine triphosphate, ATP. That's the thing that does all the work. Glucose is just producing ATP. We'll go into that later. As an energy source for the cell. In this comprehensive guide, we will explore step-by-step bioenergetics of fast glycolysis, covering each reaction and its significance in detail. So glycolysis is the first step of glucose metabolism. It takes place in the cytoplasm of the cell. That's the gooey innards between the cell wall where all the little organelles float around. So it's not inside any of the organelles. The second part is. It is a common pathway for both aerobic and anaerobic respiration, serving as the initial step in breaking down glucose to produce energy. Fast glycolysis specifically refers to anaerobic version of glycolysis that occurs when oxygen is scarce or when cells need energy rapidly, such as during high-intensity exercise. Now that anaerobic just means without oxygen, but there's oxygen bumping butts around in there, so that's kind of, as an exercise physiologist, I use the term fast glycolysis and anaerobic glycolysis interchangeably, but I really try to prefer to say fast glycolysis because it's more scientifically accurate, but you could use either. The primary goal of fast glycolysis is to generate ATP and NADH, that's nicotinamide adenine dinucleotide, which can be used as an energy source in the absence of oxygen. The process begins with the one molecule of glucose and eventually results in the production of 2 molecules of pyruvate along with a net gain of 2 molecules of ATP. Now that net gain is after all the costs of ATP. It costs to bring the glucose into the cell and to do all of this other stuff. So you get 2 molecules of ATP, 2 net gains. You produce a little bit more. And the 2 molecules of NADH, short for nicotinamide adenine dinucleotide, NAD plus in its reduced form, so I'm just going to say NADH and NAD plus from now on because that's a hard word. It is a crucial coenzyme involved in various metabolic reactions within living cells. It plays a fundamental role in cellular respiration, acting as a carrier of high energy electrons generated during the breakdown of carbohydrates, fats, and proteins. This bioenergetic pathway can be broken down into a series of 10 enzymatic reactions, each with its own unique role in the overall process. So step 1, hexokinase reaction. There's an enzyme, hexokinase, substrate, glucose, we mentioned, and the product it's going to produce is glucose 6-phosphate. So in this first step of fast glycolysis, glucose is phosphorylated to glucose 6-phosphate. This reaction requires the input of 1 molecule of ATP. So we talked about that net gain. It costs some to bring the glucose into the cell, which is hydrolyzed to ADP. So adenine triphosphate, adenine diphosphate takes one of those phosphates off, providing the necessary energy for the phosphorylation. This initial phosphorylation is a crucial regulatory step and serves to trap glucose in the cell. So it's like a restraining bolt for a droid from Star Wars. Like once it gets that phosphate on it, that glucose can't exit the cell bouncing around. It's preventing its passive diffusion out of the cell. Step 2, phosphoglucose isomerase reaction. So there's an enzyme, phosphoglucose isomerase. Anytime you see that ACE at the end of the word, it usually is an enzyme, but don't quote me on that. The substrate is glucose 6-phosphate, so what we got last time. The product is fructose 6-phosphate. So in this step, glucose 6-phosphate is isomerized to fructose 6-phosphate. The conversion from glucose to fructose represents an isomerization reaction, maintaining the molecule's phosphorylation and energy commitment. This step is essential in preparing glucose for further metabolism. Step 3, phosphofructose kinase, PFK1. Now this is a cool one. I like this enzyme. This enzyme is super important. So the enzyme phosphofructose kinase, substrate is fructose 6-phosphate, what we ended up with. Product is fructose 1,6-biphosphate. Phosphofructose kinase is a key regulatory enzyme in glycolysis. In this irreversible and rate-limiting step, fructose 6-phosphate is phosphorylated to form fructose 1,6-biphosphate. This reaction consumes 1 molecule of ATP, so there's another net cost of ATP, and commits the substrate to further glycolytic breakdown. PFK, phosphofructose kinase, I'll just call it PFK, plays a vital role in controlling the overall rate of glucose, responding to the cell's energy needs. This is the rate-limiting step enzyme for fast glycolysis. Its ability to perform is reduced when pH drops in the presence of lactic acid. So this is what happens when you feel the burn and you can't lift anymore, you can't run anymore, you can't do anything. This is what is being hindered first. So going on, step 4. Aldolase reaction. So the enzyme aldolase, again you see that ACE at the end. Substrate, fructose 6,1,6-biphosphate. So that's what we ended up with last time from PFK. Now the products are dihydroxylacetone phosphate, DHAP from now on, and glycosylceride 3-phosphate, G3P. Aldolase catalyzes the cleavage of fructose 6,1,6-biphosphate into 2 3-carbon molecules, DHAP and G3P. So it cuts that 6 molecule in half. These 2 molecules can freely interconvert as they are isomers of each other. So this is like a first cleavage, right? The first lyse, first big lyse. It's really cutting that carbon chain into 2 3s. So step 5, triose phosphate isomerase reaction. The enzyme triose phosphate isomerase. Substrate is DHAP. Let's try saying that again. Dihydroxyacetone phosphate, and the product is glycosylceride 3-phosphate, G3P. So it's going between those two because they're isomers of each other. This is going to commit to one of those. Triose phosphate isomerase mediates the conversion of DHAP into G3P. This reaction is vital because it unifies the glycolytic flow pathway, allowing the substrate reactions to produce with a common intermediate, that's G3P. Glycosylceride 3-phosphate. So step 6, glycisaldehyde 3-phosphate dehydrogenase, GAPDH reaction. So we'll go between those 2 big words in the acronym there. The enzyme glycosylceride 3-phosphate dehydrogenase. The substrate is G3P, and the product is 1,3-biphosphoglycerate 1,3-BGP and NADH. So this step involves the oxidation of G3P to produce 1,3-biphosphoglycerate 1,3-BPG, while also reducing NAD plus to form NADH. We talked about those molecules earlier. This reaction is an example of substrate-level phosphorylation as it generates 1 molecule of ATP. So we're starting to produce. It's been costing us ATP. Now we're starting to produce it by transferring a phosphate group from the substrate to ADP. This newly generated ATP is the first net gain of ATP in the glycolytic pathway. Step 7, phosphoglycerate kinase, PGK reaction. The enzyme phosphoglycerate kinase. Substrate is 1,3-biphosphoglycerate 1,3-BPG. The product is 3-phosphoglycerate 3-PG. In this reaction, 1,3-BPG is converted into 3-phosphoglycerate 3-PG, while transferring the high-energy phosphate group to ADP, forming ATP. This is the second net gain of ATP in glycolysis. Step 8, phosphoglycerate mutase reaction. So you got an enzyme, phosphoglycerate mutase. Substrate is 3-phosphoglycerate 3-PG. The product is 2-phosphoglycerate 2-PG. So you see what we're kind of doing. We're slicing off those phosphates, I think. Phosphoglycerate mutase catalyzes the conversion of 3-PG to 2-phosphoglycerate 2-PG. This reaction is reversible isomerization that prepares the substrate for the subsequent step, which is step 9. Enolase reaction. So the enzyme enolase, the substrate is 2-phosphoglycerate 2-PG. What we ended up with, the product is phosphoenolpyruvate, PEP. So instead of 2 phosphates, you just get 1, right? So the enolase facilitates the conversion of 2-PG to phosphoenolpyruvate, PEP, which involves the removal of water from the substrate. This reaction increases the molecule's energy content and prepares it for the final step. Step 10, the pyruvate kinase reaction. Pyruvate kinase is the enzyme. Substrate is phosphoenolpyruvate, PEP, and the product is pyruvate and ATP. The last step of glycolysis is catalyzed by pyruvate kinase. This product produces pyruvate. So during glycolysis, glucose, a 6-carbon sugar molecule, undergoes a series of enzymatic reactions and is eventually converted into 2 molecules of pyruvate. This conversion involves the breakdown of glucose into smaller, more readily usable molecules, along with the production of ATP and NADH. Those NADHs are going to be super important when it comes to slow glycolysis or aerobic metabolism. So pyruvate is a versatile molecule that plays various roles in metabolism. In the presence of oxygen in aerobic conditions, pyruvate is further oxidized in the mitochondria. We'll go into that later. It enters the mitochondrial matrix where it's converted to acetyl-CoA. In this process known as pyruvate decarbonylation, acetyl-CoA then enters the citric acid cycle, the Krebs cycle. Again, we'll go over this in the second video because I'm just kind of touching about what happens next, why we produce pyruvate, what's going to happen to it, because that only produced 2 ATP. We need a lot more. So acetyl-CoA then enters the citric acid cycle, Krebs cycle, to generate more ATP through the oxidation phosphorylation, so lactate fermentation. In the absence of oxygen or under anaerobic conditions, pyruvate can undergo fermentation. For example, in muscle cells during intense exercise when oxygen availability is limited, pyruvate is converted into lactate. We've heard that, lactic acid, right? As a way to regenerate AD plus from ADH, allowing glycolysis to continue and produce ATP even without oxygen. That also helps with gluconeogenesis. Under certain conditions, such as fasting or low carbohydrate diets, pyruvate can be converted back into glucose through a process called gluconeogenesis. This occurs primarily in the liver and helps maintain blood glucose levels when glucose availability is low. This has been a short breakdown of fast glycolysis. I'll do a video soon, hopefully, on the slow aerobic metabolism, where pyruvate, once converted into acetyl-CoA, goes on from here. That's some real stuff. Lightning in a bottle kind of happening in the mitochondria.